Usually, when we talk about a major security system failing, there is this expectation of a really catastrophic, explosive physical breach, like a bank vault.
Right. You imagine the heavy steel door just being blown off its hinges, alarms blaring, debris everywhere.
Exactly. The damage is undeniable. You expect to see the exact point of structural failure.
You're looking for a giant hole in the wall.
Yeah, but when you look at the human brain, specifically the blood-brain barrier, which is, it's the ultimate biological security system designed to keep the toxic environment of our blood from touching our extremely fragile neurons, the failure doesn't happen with an explosion.
No, not at all. As we age, that barrier degrades in a way that is profoundly subtle.
It really is a masterclass in microscopic infiltration. And the wild thing is, for decades, science had the fundamental mechanism of that infiltration completely backward.
We were essentially looking for a broken door when the real threat was sneaking in through a completely different, highly organized mechanism.
So, welcome to this deep dive. Today, our mission is to pull apart this absolutely groundbreaking 2026 research paper. It was published in the journal Neuron.
Yeah, the study is titled TGF-β1-induced endothelial transcytosis drives blood-brain barrier leakage during aging by Fang and colleagues.
Right. And we are going to figure out exactly how the brain's shield actually fails as we age, why the old structural theories completely miss the mark, and we're going to reveal a highly active cellular process that might amazingly actually be reversible.
The source material here is dense. We are navigating genetic sequencing, electron microscopy, molecular signaling cascades.
It's heavy stuff.
It is, but our goal is to translate that granular science, the literal molecular machinery of aging, into clear, actionable insights for you listening without, watering down the brilliant methodology these researchers used.
Because the implication here completely changes the paradigm. Cognitive decline, brain fog, neurological aging, they might not be this inevitable passive wear and tear process we always thought they were.
No, it appears to be a specific molecular switch that accidentally gets flipped in midlife.
And scientists have just figured out the exact chemical sequence required to turn it off.
Right. But to start fixing a leak, you first have to pinpoint exactly when the integrity is compromised.
Which brings us to the timeline. Because before we can fix it, we need to know when the wall actually cracks.
Exactly. The long-held assumption in neurology was that the blood-brain barrier only really breaks down in extreme old age, concurrent with severe dementia or late-stage neurodegeneration.
But the data from this paper maps a completely different timeline.
It does. The researchers mapped this by testing standard lab mice across 4 distinct life stages. They looked at young mice at 2 to 4 months old, mature adults at 6 to 8 months, middle-aged mice at 10 to 12 months, and finally aged mice at 16 to 18 months.
Okay, so a pretty comprehensive lifespan.
Very. And to visualize the integrity of the barrier, they literally injected fluorescent tracers directly into the animals' bloodstreams.
Just to see what would glow, basically.
Right. And they used molecules of varying sizes to see exactly what could slip through. They started with a tiny glowing molecule called 3-kilodextrin.
And just to clarify for you listening, a kilodalton, or K a day, is a measure of molecular mass. So 3 kilo a day is incredibly small.
Super small. But they also tracked naturally occurring massive proteins in the blood, like albumin at 66 kiloday and IgG, which is a massive 150 kiloday antibody.
So using immunofluorescence, they light up the brain to see if any of these glowing molecules made it past the barrier.
Yeah, and they found that the leakage actually begins in mid-adulthood.
Wait, mid-adulthood?
Yes. At just 6 to 8 months old in these mice, the barrier is already failing, and it starts specifically in the hippocampus.
Wow. Okay, so that midlife onset is the first major paradigm shift.
Definitely. And the nature of the leak is highly progressive and size-dependent. Think of it like a micromesh sieve or a coffee filter that is slowly stretching out over years.
I can picture that.
Yeah, in that midlife stage, only the tiny solutes, the 3-kilodextrin, and a bit of the albumin are slipping through.
Right, because the holes aren't that big yet.
Exactly. The massive proteins, those 150 kiloday IgG molecules, they don't breach the barrier until much later, around 10 to 12 months, when the system is severely compromised.
So if you're tracking your own midlife timeline, the process starts way earlier than the outward symptoms of cognitive decline. You wouldn't even feel it happening.
You wouldn't. And there was another variable they controlled for that I found really fascinating. They explicitly compared male and female mice at these critical onset stages.
Checking both the hippocampus and the cortex.
Right.
Oh.
Because often in aging studies, you expect to see some variation based on sex, perhaps a degree of neuroprotection offered by estrogen or, other hormonal factors.
But the progression of the leak, the onset timing, the severity, it was totally identical.
Yeah, completely identical. The vulnerability is entirely gender neutral.
Which points to a really fundamental, universal mechanism of biological aging, rather than just some hormone-dependent anomaly. Okay, so let me do a quick recap here to make sure I'm visualizing this checkpoint accurately, and for you to keep it straight. We were looking at a progressive, size-dependent leak that begins in midlife, affects males and females equally, and starts right in the memory center, the hippocampus. Spot on. But here's my question. If the barrier is acting like a stretching sieve, selectively letting small things through first, what is physically stretching? Are the walls themselves actually crumbling?
Well, to answer that, we have to look at the actual cellular architecture. The researchers used transmission electron microscopy, or TEM.
And TEM doesn't just use light, right? It shoots a literal beam of electrons through an ultra-thin specimen.
Right, which allows you to see the physical borders between the brain and the blood and massive magnification. They focused on the endothelial cells.
Those are the tightly packed cells that line the interior of the brain's blood vessels.
Exactly. Now, the classic scientific consensus was that aging caused the breakdown of something called tight junctions.
Okay, so if you picture the endothelial cells as bricks in a wall, the tight junctions are the mortar holding them together.
Perfect analogy. This is the paracellular route, the microscopic space between the cells. The assumption was that the mortar degrades as you get older, the bricks pull apart, and plasma just seeps through the cracks.
I mean, it makes intuitive sense. Things wear out.
It does.
Yeah.
But when the researchers used the electron microscope to examine the specific proteins that construct these tight junctions, proteins like claudin-5, occludin, and Z01, the mortar was completely intact.
Wait, completely intact, even in the older mice?
Yes. In young mice, middle-aged mice, and even aged mice at 18 months, those tight junctions were sealed completely shut.
Oh, wow. So they only saw the tight junctions physically degrade in what? The ultra-aged mice?
Right, mice that were pushing 27 months old.
So if the mortar between the bricks is solid for almost the entire aging process, the enemy isn't squeezing through the cracks.
Nope. The blood must be moving straight through the bricks themselves.
Okay, I want to use a visual analogy here to lock this in for you listening. Imagine a heavily guarded border wall. The old theory assumed the wall was crumbling. This paper proves the wall is actually fine.
The wall is fine, but the border guards themselves, the endothelial cells, have suddenly started running an illegal microscopic ferry service.
Yes, purposefully shuttling toxic cargo straight through the barrier.
Exactly. It's the transcellular pathway. The leak is happening straight through the interior of the endothelial cells via a process called caveolar transcytosis.
Caveolar transcytosis. Break that down for us.
So that ferry service relies on vesicles called caveolae, which literally translates to little caves.
Little caves. I love that.
Yeah. Under the electron microscope, you can literally watch the cell membrane physically pinch inward on the blood side. It creates this tiny bubble, swallows a pocket of blood plasma, pinches off.
And then just floats all the way across the inside of the cell.
Yes, it floats across and fuses with the brain side membrane to dump the plasma directly into the neural tissue.
That is wild. And they proved this by injecting an enzyme called HRP into the blood, right?
Because under the electron microscope, the HRP turns incredibly dark. So they could literally watch these tiny caves filling up with black dye and ferrying it across the cell.
But wait, this is where the logic hits a wall for me. Forming vesicles and moving them across a cell, that requires a massive amount of ATP. It's highly energy intensive.
Very energy intensive, yeah.
Cells don't just expend that kind of energy to sabotage their own environment for no reason. Why on earth is the brain actively financing a ferry service that destroys it?
That is the core mystery here. It isn't passive wear and tear. It is an active betrayal.
Right, so let's do another quick recap for you listening. Tight junctions, the mortar innocent. They're fine. The real culprit is increased caveolar transcytosis, the cellular ferry service. So who is giving the order to start the ferries?
To find that out, the researchers had to trace the molecular chain of command. They looked at the proteins regulating the cell membrane and found two key players fighting for control, MSSD2A and CAV1.
MFSD2A and CAV1. Okay, so MSSD2A is essentially the brake pedal, right?
Exactly. It's a lipid transporter that lives in the cell membrane and actively suppresses the formation of these vesicles. As long as MFSD2A is highly expressed, the ferries stay docked.
But the data shows that during aging, the levels of MFSD2A just plummet.
Right. And as the brake pedal disappears, the engine revs up. That engine is CAV1.
And CAV1 is the actual structural protein that forces the cell membrane to curve and form those little caves, right?
Yes, the builder protein. So in an aging brain, you have a catastrophic drop in the suppressor, MFSD2A, and a massive spike in the builder, CAV1.
But proteins don't just vanish. Something has to give the initial order to suppress MFSD2A.
Right. To find the mastermind, they screened a variety of secreted factors and zeroed in on a highly specific signaling protein called TGF-β1.
TGF-β1.
Yes. Its levels increase significantly in both the blood and the brain during early aging. And when that protein comes into contact with the endothelial cells, it triggers a devastating chemical cascade.
Okay, let's trace that cascade because the mechanism is just fascinating. TGF-β1 floats up to the cell and binds to a receptor on the outside called TGF-br2.
Right, the receptor catches it.
And once that receptor catches the signal, it wakes up a messenger pathway inside the cell called the SMAD24 pathway.
And I want to emphasize, the researchers didn't just guess this sequence. They mapped it using incredibly precise genetic tools.
Oh, like ATAC-seq, right?
Yes, ATAC-seq, which stands for Assay for Transposase Accessible Chromatin.
That sounds incredibly complicated.
It is, but essentially ATAC-seq scans the cell's DNA to find regions that are physically unspooled and, open for business.
Got it.
Then they used ChIP-qPCR, which acts like a molecular fingerprint, to track exactly where proteins are physically binding to that open DNA.
So they could actually see the binding happening. And those tools proved that the activated SMAD complex physically travels from the receptor straight into the cell's nucleus and binds directly to the promoter region of the MFSD2A gene.
Yes. It literally grabs the DNA and hits the off switch, stopping the production of the good guy.
It is targeted genetic suppression. That's terrifying.
It really is. And the implications of this go far beyond just the brain. Because TGF-β1 is a circulating factor, this perfectly links systemic whole-body aging to localized brain aging.
And they ran an experiment to prove that systemic link, which is honestly a bit unsettling.
Yeah, they took young, completely healthy mice and injected them with a specially designed adenovirus.
And the virus's only job was to make the mouse's body overproduce TGF-β1 in the circulating bloodstream.
Right. And even though these mice were chronologically young, flooding their blood with that single signaling protein perfectly recreated the aged blood-brain barrier phenotype.
So the body's circulating blood is basically flooding the brain's receptors with this TGF-β1 signal, tricking it into lowering its defenses.
Tricked it completely. The receptors caught the TGF-β1, the SMAD pathway turned off MSSD2A, CAV1 went into overdrive, the ferry service started, and the young brains began leaking exactly like the aged ones.
Wow. Okay, let's summarize this molecular crime scene before we move to the solution. The structural glue is fine. The leak is caused by cellular vesicles. The vesicles are built because MFSD2A is turned off, and MSSD2A is turned off because systemic TGF-β1 orders the nucleus to shut it down.
That is the exact chain of events, which brings us to the intervention.
The rescue. Because if we know the exact molecular blueprint of the breakdown, we can theoretically engineer a way to intercept the signal, right?
Exactly. The researchers tested this at multiple points along the pathway to see if the barrier could be rescued, and they utilized both genetic and pharmacological hacks.
Let's look at the genetic tools first. They use viral vectors, which are essentially hollowed-out viruses used as microscopic delivery trucks to drop off genetic instruction.
Right. One vector they used was AAV9, carrying a short hairpin RNA designed to knock down the production of CAV1 in aged mice.
So if you remove the structural protein CAV1, the cell physically cannot build the vesicles. The ferry service just halts.
Precisely. They also used a highly sophisticated vector called AAV-BI30. This one was engineered to bypass other tissues and specifically target brain endothelial cells, dropping off instructions to force the cells to start producing MFSD2A again.
Pumping the brakes again. And they even went as far as genetically modifying a group of mice to completely lack the TGF-br2 receptor on their endothelial cells.
Right, because if the cell doesn't have the receptor, it can't hear the TGF-β1 signal at all.
And the crazy thing is, in every single one of these targeted genetic interventions, the aging leak was completely stopped.
Completely stopped. But, viral gene therapies and receptor deletions, they are incredible proof of concept tools, but they aren't exactly ready for a quick human prescription at your doctor's office.
Yeah, a bit too complex for everyday clinical use right now.
Exactly. So, crucially, the researchers also tested a pharmacological intervention. They used an orally administered small molecule drug called LY210-9761.
Okay, an oral drug. That's much more practical. LY210-9761 is a dual inhibitor, right? Its job is to physically block those TGF-β receptors on the outside of the cell.
That's right. And they gave this drug to aged 18-month-old mice for just 7 days.
Just 1 week.
1 week of oral treatment was enough to block the systemic signal. Without the constant TGF-β1 suppression, the cells naturally turned their MSD2A production back on.
The brake pedal returned.
Yes. The vesicles disappeared and the barrier physically resealed itself.
That is amazing.
But the structural repair isn't even the most important part of this finding. They needed to know if sealing the leak actually rescued the brain's function.
Right, because the neurons are already dead. Sealing the wall doesn't matter. You're just closing the door to an empty house.
Exactly. So they put the treated aged mice through behavioral testing, starting with the Y maze.
And the Y maze tests spatial working memory, for those who don't know. A mouse is placed in a maze with 3 arms shaped like a Y. A healthy mouse is naturally curious and will explore all 3 arms sequentially, remembering which ones it just visited.
But an aging mouse with memory deficits will just keep wandering down the same arm over and over. They forget where they've been.
Right, but after the treatment.
The aged mice treated with the inhibitor made significantly more spontaneous alternations. They remembered their environment.
Oh, wow.
They also ran a novel object recognition test, placing a familiar object and a brand new object in the enclosure.
And the treated mice spent considerably more time investigating the new object, which is a massive indicator of restored curiosity and cognitive function. Rebuilding the vascular barrier literally made the older mice sharper.
It really did. This completely shifts our understanding of cognitive decline. We spent decades viewing age-related memory loss as this one-way street of irreversible neuronal death.
Yeah, that's what we were all taught.
But this data implies that a significant portion of that cognitive fog is actually driven by active reversible vascular leakage. Because it is controlled by a cellular signaling pathway, it can be chemically interrupted.
Intervening at any point in the TGF-β1 and MFSD2A pathway can rebuild the barrier and actually rescue cognitive function in older age. It's a totally new way to approach neurodegeneration.
Absolutely.
Think about the scale of what we just explored today. We zoomed out to the macro level, looking at a midlife decline in working memory, and then we pushed all the way down into the microarchitecture of the brain.
Past the tight junction.
Right, past the tight junctions, looking at individual cellular vesicles ferrying plasma, all being orchestrated by a rogue protein signal shutting down a single gene.
It perfectly illustrates how granular foundational science, tracking glowing dextrin and mapping open chromatin in a mouse's endothelium, how that eventually paves the way for very real, actionable therapeutics in human medicine.
It really does. But before we wrap up, there is one final, highly provocative detail buried in the text of this study that I want you, listening right now, to mull over.
No, I know exactly what you're going to bring up.
The researchers noted that while TGF-β1 increases locally in the brain, it also spikes heavily in the circulating blood early in aging. And they pointed out that the predominant reservoir of latent TGF-β1 in the human body is actually our platelets.
The cellular fragments responsible for clotting our blood.
Yes. And clinical data shows that as human beings age, our platelets become increasingly hyperactive.
They do.
So if a signaling protein released by hyperactive platelets in the systemic blood is what ultimately commands the brain's security system to collapse, could the future of treating neurodegenerative diseases has nothing to do with treating the brain itself?
That is the big question.
What if the key to protecting our memories and maintaining youthful cognition lies entirely in filtering or calming the systemic blood supply before it ever reaches the neck? Think about the implications of that the next time you go in for a simple blood panel.
It radically redefines where the brain's vulnerabilities actually lie.
It changes everything. Thank you for taking this deep dive with us into the microscopic architecture of your own mind. Until next time.